Good evening, everyone. Welcome to Karolinska Institute. My name is Gustav Nilsson, and tonight it's my pleasure to introduce Dr. Jordi Riba.
Should I start?
Dr. Riba is a neuroscientist and pharmacologist from Spain who is going to tell us tonight about his research on ayahuasca. Please go ahead.
Okay, thanks. So, first of all, thanks to all the organizers for inviting me to come to Stockholm. It's my first time in Sweden, and I hope you enjoy the presentation I've prepared. So, I've entitled it Ayahuasca from the Amazon to the Clinical Research Laboratory. You may wonder whose research laboratory and where. So, this is where I'm based. This is Barcelona, and I don't know if you can see the white spots on the top of the mountains there. This is northeastern Spain. We have the beach, but believe it or not, we also have snow. So at this time of the year, you may choose to go to the beach and stay for a while at 15 degrees plus, or you can go skiing. So it's a really nice spot. I recommend you that you come and visit sometime. And if Barcelona is the crown, one of the jewels is our hospital. This is where my lab is. This is Sant Pau Hospital in Barcelona. It's a very old institution. I recently celebrated its 600th anniversary, and this is one of the historic buildings. So it's definitely worth coming and strolling and having a walk in this area. So, in my lab, we have a series of lines of research. I'm especially interested in substances that interact with human perception.
So I work basically or mainly studying psychoactive substances, and I've done a lot of work on 5-HT2A agonists. These are the serotonin 2A agonists, the classical psychedelics, and basically dimethyltryptamine and ayahuasca. More recently, we have also studied kappa opioid agonists. I don't know if you've heard
about salvinorin A. We've done some studies in healthy volunteers and published some data already, and we're about to analyze and publish some neuroimaging and neurophysiological data as well. We've also done work with cannabinoids. Most of these studies are in healthy volunteers, but I also collaborate with the psychiatry department in my hospital, and especially in the field of addiction. I've done neuroimaging studies in patients who suffer from drug dependence, from cocaine dependence, who have problems with cannabis use, also with treatment-resistant schizophrenic patients who have permanent hallucinations, and we are starting now a new line of research to try to treat people suffering from psychological trauma in a different way. So, and also, I do collaborate with the neurology department in Parkinson's disease patients who develop neuropsychiatric complications, and also Huntington's disease. So I have this unique possibility of being a basic researcher who doesn't work with animals, works with healthy volunteers, but also has the opportunity to see patients, see some medical conditions that might benefit from substances that are there in the nature, that are psychoactive, that we can study, we can see which effects they have on the brain, and potentially one day perhaps we could use in a clinical setting. In the last 3 years, we've also started a very productive collaboration with the Beckley Foundation. This is Amanda Fielding. She's the president of the Beckley, and some of our most recent research has been funded by this institution. And also, I collaborate with this center. They do cell biology, molecular biology in Madrid at the Spanish National Research Council. And some in vitro data I'll be showing you today comes from a collaboration, and basically the experiments have been conducted in their lab. So my presentation outline will be the following. I'll tell you about psychedelics and ayahuasca, then what the acute effects of ayahuasca are on blood flow measured using neuroimaging techniques, electromagnetic brain oscillations, EEG, then post-acute effects. This is quite recent. We have a paper under review. It still hasn't been published. So these are psychological after effects, and then MRI spectroscopy and resting state functional connectivity. and some amazing new stuff, I think, from my point of view, for someone who doesn't work in molecular biology. It really stunned me, the results I got from this lab in Madrid, and this I'll leave for the end. So I'll mention clinical studies, some results we already have in depression, future studies we want to conduct in other disorders, and in the end, the amazing stuff. So what are psychedelics? Maybe you all know about this, but perhaps some of you don't, so I'll do like a brief introduction about this. They are substances that you can find readily in nature, in many plant species. You have these simple substances with very simple chemical structures that interact with the human brain in a very profound way and very unusual way. So this would be mescaline. You have the formula here. It comes from a cactus that grows in the southwestern desert of the United States and in northwestern Mexico. It's the peyote cactus. Then you have the magic mushrooms from the psilocybe genus. You have psilocin. This is the substance that's formed in vivo when you ingest psilocybin. Maybe psilocybin is better known. This is psilocin, what's formed in our body. And in many plant species, you have this compound. It's called dimethyltryptamine, or DMT. And it's really, really widespread in nature. And this substance plays a very prominent role in ayahuasca. But you can find it in the seeds of this other plant and in many others. So these are, as you see, very simple compounds, nothing special about them. But for some reason, they have been used since ancient times by humanity due to the psychoactive effects they produce. They produce effects in the human brain in a way that has amazed our ancestors since they lived in caves. And you can see in these paintings here in Algeria, these are from caves in Algeria, people carrying mushroom-like plants in their hands and having these widely open eyes that perhaps transmit the idea of amazement, and perhaps this is due to the plants that they may have consumed. So this is from the Sahara, but then you can go to northern Chile, and you have these burials in a very dry place, one of the driest places on earth. This is the Atacama Desert. You have these mummies. They have been they have done excavations, the archaeologists. They have recovered these mummies, and these mummies have been buried with bags that carry these seeds, which I showed before, and that contain this substance, dimethyltryptamine, or DMT. And alongside these bags with the seeds, they found all this paraphernalia, all these tablets, and all these tubes that are prepared to inhale the powder that's obtained from these seeds. So how important were these seeds to these people, this plant, that they buried their dead alongside all these utensils, all this plant material, all these artifacts that you need to take the substance. So it's obvious for us that it played a very important role in the world vision and probably the religious beliefs of ancient cultures. So classical psychedelics, all these substances I showed you, if you ask basic pharmacology what they are, they will tell you that they are 5-HT2A agonists. That means that they interact and they activate the so-called 2A receptor of serotonin. So this would be the molecular structure of serotonin. This is an endogenous neurotransmitter. We have this physiologically in our brains and outside the brain as well. And here you have the structure of psilocin. If you see, they are almost identical. There's just some slight changes in the molecule, so you can start thinking that all these substances are going to interact with serotonergic neurotransmission. And this is precisely what they do. They activate this 2A receptor, and they do this selectively. Serotonin activates this receptor as well, but also many others. So usually we are not having mystic experiences, we are not having visions while we are awake at least, but if you take psilocin, you will start having this. And this has puzzled researchers for many years, and we still don't have the answer. Why is that? That if we activate the 2A receptor, you get all these psychoactive effects. But the fact is that they do. So what we know now, what we've known for 50 years already or more, is that they activate in a selective manner this 2A receptor, and this 2A receptor is thought to be mediating all the psychoactive effects of classical psychedelics. So that was if you ask a basic pharmacologist what's a psychedelic, but if you ask the average citizen in our societies what a psychedelic is, they probably, what comes to their mind is something like this. So, yeah, it's something that you tell and you start to hallucinate. So maybe the doc gave you something and said, oh, I might produce some visual side effects. So this is the general idea that's out there in the public, but there's something more to it. It's not so frivolous, and this would be like a superficial vision of these substances. So we see that if our ancestors revered these substances, it was for some reason. And the reason is that they interact with the most intimate aspects of our psyche. And what are these intimate aspects of our psyche? Well, it's perception, the way we perceive reality, it's cognition, the way we think, and it's emotion, the way we feel. And what do they cause if you take one of these substances? Well, it causes perceptual modifications. Reality appears different, you see it different, you hear it differently, you believe it's different. There might be crosstalk between the different perceptual modalities, that's what's called synesthesia. You have increased thought speed, you have more associations. These new thoughts that come to your mind can be experienced as revelations. Perhaps this is one of the reasons why our ancestors revered these substances. And they can bring to your mind some interesting memories. And this is something that will have importance in what I'll tell you later. So in the emotional sphere, you see also increased effect. Maybe you are feeling nothing before you take one of these substances. After 2 hours, you might be crying for some reason. So there's emotional ability. It's easy to go from experiencing elation to experiencing very intense sadness. And you can experience opposed feelings simultaneously. So it's quite crazy if you think about it. So the sense of self also becomes blurred. And what is generated, if you take all these into account, is a modified state of consciousness. Your vision of yourself and of reality is changed. And all this resembles, if you have to compare it to some experience that's common to all humans, to dreams. So that's the most usual metaphor. So as you see, the effects are far more complex than what one might think if you just simply ask a sensationalist journalist what psychedelics are. So you might think, okay, this is all very well, very interesting, but should we study this? Is this like a legitimate field of research? And someone might come up with reasons why we shouldn't study psychedelics. The most immediate that comes to everybody's mind is that this is hippie stuff. So why should we be studying hippie stuff? We know that that was a failure. It was in the 60s. People got crazy, took drugs, etc. Whereas we are serious scientists. So why should we mix with those crazy people with long hair and wearing those glasses and flowers in their hair? And this is my personal answer to this question. And here's my reason why we should study psychedelics. And the answer is precisely because we are serious scientists. And if you have a substance out there in nature that can cause all that I showed you before, if we are not studying this, we are idiots, right? Because you have simple chemical structures, very complex brain, they interact, they can change the brain, how the brain works in a very profound manner, and we simply want to ignore this because someone abused this 50 years ago. Isn't this crazy, don't you think? Okay, if you are scientists, we are curious. That's the definition, or used to be the definition. Now scientist is someone who wants to advance a career, get published, get power, maybe get a position, a chair, 200 papers in some top journals, whatever that might, if that's worthwhile, maybe. But I think if you are a scientist, you have to be curious, right? So that should be the initial step. And then there's a series of data that show us that these substances might have interesting and positive effects. It's not all craziness and psychosis and hippiness, right? Might be some data that you obtain for animals, and you see that in animals, the substances increase cognitive flexibility, they increase associative learning, they show antidepressant responses when you administer these substances, and depression is quite frequent in our societies, isn't it, right? So why not look into this? Then you see that they promote some brain factors, like brain-derived neurotrophic factor that stimulates brain plasticity. So this is interesting as well. And there's a renewed interest in the substances. So some initial studies have been carried out in the last 15 years or so, showing that they can increase well-being and provide meaningful experiences to people who are healthy, but also to people who are suffering. People who are suffering from depression, people who are suffering from the anxiety associated with terminal illnesses like cancer. So this has proved that it's effective. Of course, this is an underfunded field. So many of these studies have methodological aspects that could be improved. But these are the initial steps, and we are going in the right direction. And I think they are demonstrated that it's reasonable and it's legitimate that we study these substances. So this is basically why I think we should study this. And fortunately, there's other people out there who share this vision. And some recent papers have appeared also in top journals for all of those interested in promoting their scientific careers, right? This is Archives of General Psychiatry. They showed that psilocybin was used to treat the anxiety associated with cancer. This was published by Charles Grob and his team in 2011. And more recently in Lancet Psychiatry. Doesn't sound like an underground journal, right? Lancet Psychiatry. Well, they showed as well, this was a study conducted in England, that psilocybin was useful for treatment-resistant depression. So now, that was an introduction for psychedelics, why we should study them, what they are. And now I'm going to tell you about ayahuasca, for those of you who don't know what ayahuasca is. So I asked myself this question in 1994 when I first heard about ayahuasca. And back then, you had to access some artifacts that they were made of cellulose, and they were called books. And you had to read those books. So they looked like something like this. If you wanted to know what ayahuasca was, you had to make some cognitive effort to extract the information from there. Things have changed in recent years. I did this morning in my hotel. I went and I Googled ayahuasca. And I got in 0.36 seconds, I got 4,160,000 responses as to what ayahuasca is. So today, information is out there, is available, and I think ayahuasca has become more and more popular. So I think it was like 3 weeks ago that I checked online, and it was only 4 million entries. Now, in 3 weeks, it's 160,000 more entries that you have regarding ayahuasca. So this is popular. So if you're curious, you go into Google, you introduce the word, you can find lots of information, but maybe you don't go into Google to introduce the word ayahuasca. And on a Sunday, you can grab the New York Times, and they tell you under fashion and style that ayahuasca is a strong cup of tea, right? So even if you're not of the sort that goes online to Google weird names like ayahuasca, dimethyltryptamine, psilocybin, etc., maybe one Sunday you get your newspaper and they tell you what ayahuasca is. So you might get curious and start looking for some place where they offer this. So what's this strong cup of tea that we see published in the papers? Well, this is an ancient psychotropic tea. It's I call it a Pan-Amazonian psychotropic tea because it's been used in the upper Amazon for centuries in countries like Peru, like Bolivia, like Brazil, in the westernmost part of the Amazon, also in Venezuela. And this psychotropic tea is obtained from this plant. This is a vine. It's called Banisteriopsis capi. It's a climbing vine, also called a liana. And I would like to point out that this is the main ingredient of ayahuasca. This vine in Quechua is called ayahuasca, and the tea that's obtained from this vine is also ayahuasca. The problem comes when many of the human groups that used this vine to obtain the psychotropic preparation added also other plants to the brew. So this basic ingredient is combined with many other plants, and the most popular that has crossed borders, that has reached Europe, that has reached North America, Japan, contains also this other plant. This is a shrub. It's called Psychotropirides. So usually when you hear about ayahuasca, they refer to this combination. And what we've studied in the lab is this combination. But ayahuasca can be 1,000 other things and 1,000 other preparations, but all of them contain the liana, the Banisteriopsis capi. So the traditional uses for ayahuasca was in shamanic healing. And so these human groups in the Amazon believe that there are spiritual causes, to illnesses. So the traditional healer or the shaman, what he did was take ayahuasca himself, not the patient. This has been interestingly forgotten in recent years. Many people want to go under ayahuasca healing experiences, and they forgot that it was the doctor who took it, not the patient, right? But anyway, we are always reworking on ayahuasca. So this was one of the main uses. It was used to diagnose and try to cure illnesses. By other human groups, it was used maybe once in a lifetime in a rite of passage. When children became adults, they had to go through an ayahuasca ritual, and they had, in some tribes perhaps, look for a spirit that would protect them for the rest of their lives, to get a vision. So that was the traditional uses. And then came some interesting syncretic movements called the ayahuasca churches that blended together, and this happened in Brazil, Christian beliefs with traditional ayahuasca use. So in this context, ayahuasca became a sacrament central to their religion. And these churches exported their activities to North America, to Europe, and this is when I got to know about this in the mid-1990s. So there were ayahuasca using groups in the Barcelona area. I thought this was quite amazing, quite interesting. Nobody knew what that was. You couldn't Google it because Google didn't exist back then. As I said, you had to read books. It took me a while before I knew what I was getting into. So I decided, I was back then doing research in the organic chemistry. So I finished that and I said, okay, I'm going to do my PhD in pharmacology and I'm going to study ayahuasca. So I did this under the supervision of the late Professor Manuel Barbanoch. He was a professor of pharmacology. He was very interested, especially in psychopharmacology. So I approached him in 1994, and I said, okay, you know, there's this weird Brazilian groups coming to Barcelona, and they're having these rituals, and they get this mixture of plants, and they take it, and they have visions. And he was the only person who didn't think I was crazy. So I was able to start doing my PhD there, and I knocked at several doors before. And they showed me their sympathy. I got a tap in my shoulder, and I had to leave. So Manel really was interested. So since then, since our first publication, I think we have been reasonably productive. Like we've published 31 journal articles, 5 book chapters. So we know a little about ayahuasca, but the road was not easy. So it was in 1994 that I had the first contacts with ayahuasca using groups. In 1996, we started this research program at Sant Pau Hospital. And then, imagine, we had to develop the ayahuasca formulation we would use in our trials, design what we wanted to assess, get approval from an ethics committee from the Ministry of Health, and we started our first study in patients in 1999. First article published 2001. So my message to you, someone who's sitting in the audience and wants to be a researcher, is you have to be persistent. And if you want to do research in this area, you have to be twice as persistent or 10 times as persistent. So it's very nice and well, and it's you get these papers and you read the abstract in 5 minutes, and maybe it took like 6 years to get things done, right? So it's not easy. So I don't know where the reward is. Maybe I should put myself in an MRI scanner and see what's wrong with my brain. But I've been working on this for 20 years now. So the chemical constituents of ayahuasca, the main focus has traditionally been dimethyltryptamine. This is because it's a classical psychedelic. It interacts with the 2A receptor. It has very intense psychedelic effects when given parenterally. This means when it's not taken orally. That means injected, smoked intravenously or intramuscularly. And why is that? Why is it inactive when you give it orally? Because it gets very rapidly degraded. You have some enzymes in your gut, in your liver, and it doesn't reach systemic circulation alive. It gets wiped out immediately. But in the Amazon, they came up with this idea of combining the leaves of this shrub with the Banisteriopsis scapi vine, the ever-present ingredient in ayahuasca. And Banisteriopsis scapi vine, the Banisteriopsis scapi vine has these alkaloids, these other substances. They have harmine, harmaline, tetrahydroharmine. These are those that are most abundant. You have others. And these substances are potent monoamine oxidase inhibitors. This is the enzyme that wipes out DMT when you take it orally. So if you ingest these substances in the ayahuasca brew together with DMT, you are going to get an interaction, a very interesting interaction. And also, tetrahydroharmine is a weak monoamine oxidase inhibitor, like some antidepressants. And traditionally, the view that pharmacology has had of these substances coming from Banisteriopsis scapi is that they play a secondary role in the overall pharmacology of ayahuasca. They are just there to allow DMT to be absorbed and to exert its effects on the central nervous system. But as I mentioned earlier, at the end of my talk, I'll show you some cellular effects, the amazing new stuff that can produce these substances, these beta-carbolines, the alkaloids you find in Banisteriopsis scapi. So when you have all these substances combined in this soup that's called ayahuasca, you ingest it, right? Well, if you took DMT alone, as I said, it gets degraded. It doesn't pass this firewall. It doesn't reach your systemic circulation, the bloodstream. It doesn't reach your brain. When you take it together with the beta-carbolines, with the harmalas in ayahuasca, DMT reaches the brain, and interesting things start to happen. So we wanted to study this in the laboratory, and we had to do it in a way in which the substances in ayahuasca would not degrade, and that we could measure the exact amounts we were giving to our participants, and that we could implement double-blind designs, simple blind designs, that we could compare the effects with placebos, inactive placebos, active placebos. So what did we do? And this took us a few years to sort out. What we did was we freeze-dried ayahuasca. Have you heard about Nescafé? Okay, we did the Nescafé of ayahuasca, right? You basically freeze ayahuasca in a chamber in which you extract all the air, so you have very low pressure, and the frozen water simply evaporates. So you are left with everything that's in the ayahuasca except for the water. So that would be the original ayahuasca tea. This would be the freeze-dried material. So this you can analyze, you can know exactly the alkaloid contents, and you can encapsulate this. So you can give it in a capsule to a participant, and you can play with expectancy, right? You can give different doses, see what the dose response pattern is. You can compare it with something that's inactive. You can compare it with something that's active, but that has another pharmacological profile. We've done this, for instance, with amphetamine. So once we got this, we did our first pilot study. So the moment of truth came. We recruited some participants. This one was wearing his favorite T-shirt for the study. He's about to ingest the ayahuasca capsules. And if I remember correctly, he voiced a concern. He mentioned that perhaps the spirit of ayahuasca had been lost in the freeze-drying process. So there was this moment of uncertainty, but luckily for us, this was not the case, right? So fortunately not, the effects were there. And if you see here, we, in this study, we administered placebo, 3 different doses of ayahuasca. You see this neat dose-dependent increase in perceptual effects. This is measured using an instrument called the hallucinogen rating scale, also the affective modifications, and also the cognitive modifications. So this is standardized questionnaire. We have these results, but if we ask the participants, okay, tell us what you've experienced, then they said, okay, this was really not recreational, and this is something that we already knew from the people who took ayahuasca in the rituals that I had attended, those who were involved in these Brazilian ayahuasca religions. So there were some introspective dreamlike experiences. This was had been reported for psychedelics, something that didn't surprise us. Visions with eyes closed. This would be typical as well for all psychedelics. But then, interestingly, there were very powerful autobiographic memories that came to mind during the experience in the laboratory context, intense emotional effects, and they said that they gained some interesting insights into personal life matters. As you see, this is not something that you would take to go to a party, right? The experience could be very intense. It could be really overwhelming. No wonder that these Brazilian groups had turned ayahuasca into a sacrament and the ritual use into a religion. So what's going on in the brain? So this is what people tell us that they experience, but maybe they are making this up. Maybe nothing's going on there. So how could we study this? So we did a study, a neuroimaging study. But first, I want to show you where these 2A receptors, these serotonin 2A receptors, are basically localized in the brain. So this is basically a cortical receptor. Here you have this table showing that you have large amounts in the calcarine cortex. That's the visual cortex, the primary visual cortex. So maybe no wonder that there's some visual effects going on. Also in the parietal cortex, you have very high concentrations of these proteins, of these binding sites for dimethyltryptamine, and also in this area in the medial frontal cortex. And these last 2 areas, maybe you're familiar with them, but they have been discovered in the recent years to be the main hubs of the so-called default mode network. Are you familiar with the concept? Have you heard about it? So the neuroimaging researchers found out that when they put people in a scanner, they typically, what they did was they imposed a cognitive task on them. They wanted to be how people process like speech, so they showed words to them, or memory, or any other cognitive function, and they compared this to the baseline situation, which is do nothing. But another group of researchers analyzed what goes on in the brain when participants are simply doing nothing. And when you're doing nothing, the brain is very active. And there are a series of brain areas that are having synchronous activity. And this is the so-called default mode network. So these 2 regions in the medial aspects of the brain are key areas of this default mode network. So when your mind is wandering, when you're thinking about yourself, when you are making plans into the future, when you are daydreaming, this default mode network is really, really active. And this has been linked to the intimate sense of self. This sense of self, this permanent voice, this continuous pattern of thoughts, of ideas that you identify with you has been associated or is thought to be related to activity in this default mode network. So as you see, these receptors are going to be in key areas of the brain. And if you administer a psychedelic and you measure in a brain slice what goes on in the region where the substance binds to the receptor, you see that when you give saline, you have a baseline activity of these neurons. When you give the psychedelic, this activity increases. So these substances in these brain slices, they activate the neurons in those areas of the brain. What they call it's excitatory postsynaptic potentials. So the neurons are working faster and more intensely. They discharge more often, right? And when you give the certain antagonists, this activity induced by the psychedelic is blocked. So it's been associated, the activity of the psychedelic, with another neurotransmitter in the brain, which is glutamate, and this is the main excitatory neurotransmitter in the brain. So there's an interaction between the 2A receptor and certain receptors for glutamate that lead to increased excitatory activity in these areas of the brain. So this is what we knew from animal studies. The previous slide I showed you where in the humans you have the highest concentrations of the 2A receptor. And then what we did was we gave ayahuasca to a series of participants, and we obtained images of the brain during the acute effects. Right? We did this using what's called SPECT, single photon emission tomography. And what we saw was quite interesting, and it matched quite well what they had reported they were experiencing during the acute effects. So we saw that there were increased blood flow, so we can associate that with increased activity in some areas of the brain that are associated with the processing of emotion and memory. So you have this area of the brain, the anterior cingulate cortex, that's at the frontier between the emotional and the cognitive. The subgenual anterior cingulate cortex is very clearly associated with emotion, and its activity is abnormal in depression. You have the insula. It's also a center for the processing of emotion. And then you have the hippocampus. That's a central structure to process memories in the brain. And amygdala, again, another structure associated with emotion. So this is what we saw under the effects of ayahuasca using this technique. We've also used another technique which involves registering the spontaneous brain electrical activity. So you have electromagnetic activity going on constantly in the brain. You can administer a drug and see how this is changed. A way of doing this is by analyze the energy in a series of frequency bands, and you can do this by recording the electrical signal. This would be EEG, or you can do this by recording the magnetic signal. It's another technique. It's called magnetoencephalography. I'm going to show you here some results from the EEG analysis. So if in the previous slide I showed you that there was something objective going on in the brain during the acute effects, which was this increase in blood flow, when we analyzed the changes in the EEG signals, we also found some interesting patterns. So when you take a SPECT image, you don't have any temporal resolution. You decide in which moment you administer the radio tracer, and later you obtain like a photo finish, like a single image of how was the brain at that precise time point. With EEG, you can take repeated images, so to speak. So here what we saw is a clear dose-dependent effect. This would be a low dose of ayahuasca. This would represent the head, the scalp. The white dots represent where the electrodes are. So when you give a low dose, you see some red spots. That means that brain electrical activity has changed. This is a probability map. It's showing where these changes were statistically significant, but it's just a few dots here and there. When you increase the dose, you see that the effects are much more intense, and also you see a time pattern. And this time pattern matches perfectly what the persons report under the effects. They say that there's a lag time. Usually the effects start around 45, 1 hour after administration. They reach a maximum between 1.5 and 2 hours, and then they gradually decrease to completely disappear at around 6 hours. So this would be just an image showing you that's, again, an objective measure that shows that something is going on in the brain. And if we analyze the different frequency bands, we see that there's a very prominent reduction in power in many of them. What's going on here is what's called desynchronization of the EEG. So it gets the slow waves decrease their power. This is basically seen in delta and theta, and especially in alpha. And you get changes in the medial frontal cortex, in those areas that also showed up in the SPECT analysis, in the neuroimaging analysis, in the blood flow study I showed you before. But now, this is a surprise, this had not showed up in the SPECT study. We saw very prominent decreases in this area where I indicated you earlier that there's a high concentration of these receptors. In this area, in this keynote of the default mode network, we are having a decrease in this brain wave, in this alpha. And alpha is an inhibitory rhythm. So if alpha decreases, it means that activity in this brain region is increasing. So we can see again what goes on, that there's something objective there. In certain brain regions, it's not widespread, it's not everywhere, it's localized in certain areas. And if we repeat the experiment, but this time we administer 2 ayahuasca doses, so we administer a first ayahuasca dose, you can see here this time point at 45 minutes, you start having the effects, they increase, they get a maximum here. And in this experiment, this particular experiment, 4 hours later, we administer the second ayahuasca dose. And the same thing appears again. So this we have reproduced many, many times, is a very robust finding. This has been recently described also for psilocybin and for LSD using magnetoencephalography. So this medial area of the parietal cortex, this central hub of the default mode network, seems to be a very specific target of psychedelics. Another experiment we did, in this case, we administered again ayahuasca, the same results for alpha, but here we wanted to see what happened if we antagonized the two-way receptor. If we administered an antagonist, a substance that binds to the receptor and prevents DMT from getting attached to this receptor. So here you have the results after ayahuasca. Here you have the results after ketanserin. This substance blocks the two-way receptor. Nothing happens when we administer it on its own. You don't see any effects. In alpha, there's no decrease, no increase, no change at all. And then when you give the combination, this ketanserin plus ayahuasca, ketanserin blocks completely this decrease you saw here before. So this is going through the two-way receptor. So this is the demonstration that in humans, this alpha reduction that you get with ayahuasca is mediated by the two-way receptor. And how is this associated with the subjective effects, what people experience? Well, we had this nice correlation. We measured the intensity of the visual effects. So when there's an alpha decrease, you have more intense visual effects. And when you administer the combination ayahuasca plus ketanserin, the visual effects were much more diminished. So also a clear link here between something that's objective, that's neurophysiology, with subjective effects. We've gone further analyzing these brain oscillations. We've introduced another technique, more sophisticated one. It's called transfer entropy. And here, what this statistical measure shows you is how information is shared between different brain regions by measuring the EEG signals and applying this formula to the different recordings. So here would be, you would have time in the x-axis, here a series of effects in the y-axis, and the yellow bars would show you the number of significant changes in transfer entropy. You see that it follows a pattern. It reaches a maximum around 90 minutes, then it decreases thereafter. This follows closely the increase in DMT plasma levels. The maximum plasma level is reached a bit before. And then you have the subjective reports. Every certain minutes, we ask the participant, how intense are the effects you are feeling? So you get a measure from 0 to 100, and you see that this matches very well what you are measuring, applying this metric, this transfer entropy. And this transfer entropy allows you to see the interaction between the different brain regions. So during the acute effects of ayahuasca, certain brain regions decrease the transfer of information, the transfer of communication between them. So there's a decrease in anterior to posterior connectivity, and you have some areas that we call the sources that are involved in this decrease, and there are some areas that we call the targets that are involved in this decrease. So that means that the frontal areas of the brain are reducing their influence, their modulation of activity in areas that are basically associated with sensory processing. So usually at the top of the neural hierarchy, you have a set of areas, basically the prefrontal cortex, that's controlling activity in most other brain regions. And here you have subjects with eyes closed. You measure brain electrical activity, and you see a reduction in front of parietal information transfer. There's a reduction in this influence from the frontal lobe towards sensory selective areas. And in addition to that, there's an increase in connectivity, but that goes the other way around. So here you have people with eyes closed, but their sensory selective areas are being the conductors in this orchestra. They have become the source areas, right? They are influencing more frontal regions. So in a way, you have reversed the way how information is processed in the brain during the effects of ayahuasca. So how come here people are with eyes closed, so there's no visual, no auditory information coming in, but these sensory selective areas are influencing the rest of the brain. They are taking over, and the frontal cortex is decreasing its cognitive control all over the brain. So we can say that ayahuasca, based on these results, modifies neural hierarchies. So there's a change in our internal dialogue. So there's a reduction in what we call, what cognitive neuroscientists call top-down control. There's an increase in bottom-up information transfer. That would be from sensory, from mnestic to your mind's eye, probably. And the way in which you process information becomes also quite different. So this an article we published very recently. Under ayahuasca, people were asked to do some creativity tests. So convergent thinking, traditional thinking, the structured way in which we find associations between items that we were presenting the participants was decreased. That was the cost associated with the ayahuasca intake. But new non-restricted ways of thinking, what we call creative or divergent thinking, emerged. So new, more creative associations emerged during the execution of these tasks, during the effects of ayahuasca. And we think that this is closely related as to this change in information exchange between brain areas. And interestingly enough, in one of our experiments, we administered ayahuasca in the morning. So many hours later, the person went to sleep in our lab, and we did sleep recordings. We divided the night into a series of bins, of cycles. And what this graph is showing you is the energy. This is a spectrum analysis of what's called slow wave sleep. Right? And the usual pattern when you have taken no substance at all, this would be the solid black line, is that you get more slow wave sleep. This is what makes you feel refreshed the morning after. Okay, you have more slow wave sleep in the first cycle, and it gets decreasing gradually in the second, in the third, in the fourth. So under amphetamine, in this study we administered as an active control amphetamine, what we saw here was a decrease in this slow wave activity. But after having taken ayahuasca in the morning, this is not immediately before going to sleep or during sleep, there was a very clear increase in this slow wave activity, right? And what does this compare to? Well, it compares to a situation in which a person has executed or an intense intellectual activity. So it's not surprising that people in these ayahuasca religions call their session trabalhos in Portuguese. That means works. So it's not recreational, really. They get exhausted after taking ayahuasca. So something very powerful goes on in the brain that leaves the person absolutely exhausted after the experience. And in the night, they sleep really, really tight. Okay. So moving on. After 20 years working in my lab in the hospital, people started reading about ayahuasca, and suddenly I got a knock on the door. That was great news. And it was a group of psychiatrists from our psychiatry department who had also been ostracized for many years because they were interested in mindfulness. So they were teaching mindfulness meditation to many of their patients because they think that the capacities that you promote by means of this training facilitate the therapeutic process in many psychiatric conditions. And they said, you know what, we think that there might be some similarities, some analogies between what we try to achieve through mindfulness training and what you describe in your papers as being the typical effects of ayahuasca. So these two interests came together, and he said, could we do a study in which we went to a group of ayahuasca-taking people, we administered the questionnaires we usually use to measure these facets, these mindfulness capacities, and see if ayahuasca is able to modulate this? And I said, yeah, okay, let's organize this. This is what we did. And we administered 2 questionnaires. I don't know if you're familiar with this. One of them is called the 5 Facets Mindfulness Questionnaire, or FFMQ. If you work in this field, this is quite well known. This is called the EQ, or Experiences Questionnaire. The FFMQ has a series of subscales. And then you have this composite index that this group in Barcelona had developed that uses those items in both questionnaires that are more sensitive to the practice of meditation. So here what you see are the pre and post scores in the different subscales of the FFMQ, in the EQ and in the mind sense composite index. And we saw that a single ayahuasca dose was able to increase the scores in these capacities. That was not during the effects, but in the 24, 48 hours, excuse me, after an ayahuasca session. So there was an increase here in this facet called non-reacting. That means the person increases their acceptance of their thoughts, of their feelings, as being something transient, not identifying excessively with, let's say, depressive thoughts, that they can put some distance between the thought and their own identity. They don't have to think that the depressive thought is them, right? They can distance themselves from that. So this is non-reacting, also non-judging. That means, okay, this is something that goes on in my mind. I think that I'm useless, I'm worth nothing, I've achieved nothing in life. Okay, I won't judge that. Okay, it's transient, I forget about this. The Experiences Questionnaire also measures what's called decentering, also getting distance from one's self-destructive thoughts. That was also increased, and this mind sense composite index was increased, and the scores we obtained were in the range that's typical for people who have gone through very prolonged periods of mindfulness meditation training. So this left them really stunned. They didn't expect this. They say, okay, it takes us many sessions to achieve this, and you're telling us that you're getting this as a single ayahuasca session. So now what we are trying to work in is see, okay, could we use ayahuasca in the context of mindfulness training, just having one or a few sessions, and see if that could work as a catalyst to accelerate this process, to make it faster, to make it more productive. This is a project we are working in. And some new data. This is currently under review. Here we combined these mindfulness measures with an MRI study. What we measure here is concentrations of metabolites in the brain. They use a technique called magnetic resonance imaging spectroscopy. It analyzes, it's like getting a chemical analysis of a certain brain area. And we saw that in that area where we had observed those decreases in spontaneous brain electrical activity, and in that specific hub of the default mode network, we see that not during the effects, this was measured in the 24 hours after the acute intake of ayahuasca, all psychoactive effects had disappeared. There was a decrease in glutamate and glutamine, in excitatory neurotransmitters. So I showed you before that animal studies had indicated that when you give a psychedelic, activity in those neurons increase. So here we didn't measure the acute phase, but afterwards, 24 hours later, what results is that levels are lower than at baseline, lower than before ayahuasca intake. So this was like an indirect demonstration that glutamate is involved in the effects of psychedelics. It has not been shown in humans yet. Also, this substance, creatine, which is associated with energy processes, and this other compound here with a very long name, I won't mention it now. This is very directly, it's directly coupled to glutamate, to excitatory neurotransmission. So you get a decrease in this, what we call a neurometabolic deactivation of this brain area. And interestingly enough, this brain area is hyperactive and associated with negative thoughts, with ruminations in depression. So this is what our spectroscopy study showed us. And in this study, we measured again mindfulness facets. So you see how they scored before the ayahuasca intake. This was in the 24 hours later, and this was at follow-up 2 months later. So levels had gone a bit down, but were still above baseline values. So we tried to see if there was any association between what we had measured using spectroscopy and these changes in mindfulness capacities, and we saw that there was indeed a correlation. So the more decrease we had in this excitatory neurotransmission in the posterior cingulate cortex, in the node of the default mode network, the higher were the scores in this non-judging subscale of the FFMQ in the 24 hours after the ayahuasca session, but also at follow-up 2 months later. So those were the results from the spectroscopy study. Also, we did a resting state functional connectivity. I don't know if you've seen these graphs before. Here you see a series of brain areas that are positively correlated. You see the warm colors, the red and the yellow here. So this means that these regions are functioning in synchrony. This is showing you the two main regions of the default mode network, and these two regions show a negative correlation with this other structure here, which is part of another network. It's called the task-positive network. So these two networks are anti-correlated under normal circumstances. So hence, the bluish or greenish colors here. This shows a negative correlation. So in the 24 hours after ayahuasca intake, this negative correlation has disappeared, right? So they are not anti-correlated. So when you compare the two situations, you see that actually these two regions, what they are doing now, is that they are acting in synchrony. So you have changed here the pattern of brain activity, not during the acute effects, in the post-acute stage. So there's an increase in crosstalk between networks that usually do not communicate, do not work in synchrony. There's an increased connectivity between them. And what does this mean? I'll tell you also in a while. Also, this other area, this anterior cingulate node, if we take this as a starting point and see how this correlates with the rest of the brain. So as we saw before, it has increased its synchrony with that posterior hub, but it has also increased its connectivity with this area of the brain. And here, what you have is what's called the medial temporal lobe, and this is an area that's associated, as I told you before, with emotion and memory. And what's this involved in? It's involved in attention. So 24 hours afterwards, you have a decrease in this area associated with negative ruminations in depression, and you have an increase in connectivity between the brain regions clearly associated with cognitive control and attention, and brain areas that are associated in emotional processing and memory. And if you take a look at how this relates to the changes in mindfulness facets, you find something similar to what we saw before. That was a spectroscopy. This is resting state connectivity. This increase between the anterior cingulate cortex and those areas of the brain processing memory and emotions correlate with increased scores in the FFMQ subscale in non-judging, and this takes place in the acute phase. But also they can predict how the scores of that person will be 2 months later. So the more correlation between the brain, these two brain regions, the higher the scores 2 months later at follow-up. So here you're obtaining some effects, you're modulating brain activity beyond the acute phase of the intoxication, and that's predictive of how this person's capacities will be 2 months later. And this is useful. This is what psychiatrists try to promote. They think there's a transdiagnostic deficit. They think that many patients having different labels may benefit if their mindfulness capacities are increased, irrespective of what the specific illness they are suffering is. This led in parallel to a first clinical study. This was conducted in Brazil. This is by a group I collaborate with. And we wanted to see if ayahuasca could have antidepressant effects in patients for which no usual medication worked. So these were people who had tried many antidepressants. They were not getting any benefit. So it was like a large last resource attempt. So we said, okay, there's nothing to lose about this. And in Brazil, this is ayahuasca is part of the cultural tradition of the country. So it's protected by law, the use in the religious settings, the use by the indigenous populations. So this was not a taboo. This could be done. And here we recruited a small sample of patients. This was just 6. And we measured the intensity of their depressive symptoms using the Hamilton Depression Scale and also the Montgomery-Åsberg Depression Rating Scale. And we did this before ayahuasca administration, a series of minutes afterwards, and then on day 1, day 7, day 14, day 21. In addition to these 2 measures of depression, we also used this scale, the Young Mania scale, that measures potential mania-like symptoms, right? So if you see here, the green line, which is the mania-like symptoms, remains unchanged along all this time until day 21. But the 2 other scales measuring the depressive symptoms show a clear and statistically significant reduction already at 3 hours after administration. And this remains decreased with a little bump here upwards, but still lower than baseline values until day 21. If you think how traditional antidepressants work, you need 3 weeks, usually, before they start showing, before the patient starts showing some benefit. And here, after a few hours, you are getting these improvements, and you don't need any additional dose. This was a single dose, and 21 days later, these people who were resistant to many other traditional pharmacological treatments are still better than they were at the beginning. So maybe you have heard about ketamine and ketamine use in depression. Are you familiar with that? Yeah. So I was talking the other day to a psychiatrist in my hospital, and I told him, if I had said to you 20 years ago that we should test ketamine in depression, what would you have told me? And he said that you were crazy, that you're crazy, right? And now this is like the top priority drug in the development pipeline of a pharmaceutical company for the treatment of depression. And with ketamine, they've seen that you also get very fast benefits and very fast antidepressant effects. So you had a substance that was used as an anesthetic and that was demonized by the press as being a drug of abuse used by irresponsible youngsters at raves, and that you could have never guessed that it could have a clinical application under that, other than putting people into unconsciousness. And now you see that they have that it has some potential beneficial effects for people, for patients who are really in need. So we believe that this might also be the case for psychedelics, at least for ayahuasca, in this case. So that this, we published this in a Brazilian journal. It was rejected by many journals because it was a small sample, it didn't have placebo, etc., etc. So it was finally accepted there, but it had a broad media impact. So Nature made a comment on our study, and we are very grateful to them because now the study, I think it's been cited by other articles around like 40 times, something like that. So it caught a lot of media attention. And I think this is getting more and more accepted, that we should look into the positive aspects of these substances. So we replicated also in a later study, and we found the same. Also very rapid and potent decreases of depressive symptoms. This was in a sample of about 16 participants. And we have some pilot data and some evidence that ayahuasca might have also anti-addictive properties. We have done field studies in Brazil of people who are involved in these ayahuasca religions. They had prior problems of alcoholism. They were really problematic drinkers, and they had given up taking alcohol altogether after starting getting involved with these groups. There's a hint there that this might work. There's been a study published in Canada also with positive results. And we have also a series of anecdotal cases of people who were very desperate and, die-hard heroin and cocaine users who were on the verge of overdosing. The next step for them was just getting an overdose and dying. They were infected with HIV, with hepatitis C, etc. And informally, they went to Brazil through certain channels, connections, etc. After a series of ayahuasca sessions there, they were able to completely give up this drug use. So we are quite impressed with those results. That's not a clinical trial. There was no placebo. There was no control group. But it hints that there might be something there. So we would really like to look into this as well in a more controlled and better design setting. But if all what we do is follow our prejudice and think that these drugs have no promise in therapy, of course you don't get the funds, and then the studies you do are with rudimentary designs, right? And then you get this review saying that, okay, this is not credible because you don't have a control group or you don't have a placebo, etc. But how can you implement that if you don't have the funding resources to do it? So gradually, I think these studies are being useful to break the taboo and perhaps bringing in some very much needed funds to do this in a proper way, and finally find out if they work or if they don't. And if they don't, I'll accept this. I'll be happy with that. I got into this field because I was interested in the mechanistic aspects of psychedelics. I wanted to study how they interact with the brain, not because I want to spread the gospel that they are very fine things, etc. But if they can help some individuals, I'm really happy to hear that. So when some former heroin addict comes to me and says that she's basically alive thanks to ayahuasca, then I think, okay, maybe 20 years of doing this research has had some purpose or some meaning. And this is, at least for me, the greatest satisfaction I've had. That's to think that this can be useful for someone. So, in addiction, going into this field, as I told you in the beginning of my talk, I work in this area, and we did a neuroimaging study in cocaine addicts. So this is something totally unrelated to ayahuasca research, but I'm bringing this up to show you that what we found in this study is there were functional differences in how people process reward and punishment in a functional test, but there were also hardwired changes in their brain structure. So what do I mean by that? There were changes basically in what's called the reward circuit of the brain. So all those areas got more interconnected. This is a measure called fractional anisotropy. It measures the integrity of white matter tracts. I'm not showing a lot of other slides I had originally thought of including here in this presentation, showing that there's also an increase in volume in all those regions that process reward. So that's like working at a high pace, but then connectivity outside this reward circuit is decreased. So these are frontoparietal tracts, white matter tracts, and this is decreased. So here the reward circuit is taking over at the expense of everything else. So we are asking people to give up their addiction, and their brain is already, perhaps due to the drug use, this is not longitudinal, I cannot say this for sure, but their brain is different. So you tell them that, okay, you should put more effort into this. But okay, I was amazed when I saw this. Okay, you're asking someone whose brain is probably already hardwired to keep on taking this. And the reality is they keep relapsing and relapsing. So how can you break this? So shouldn't we look at the drug that apparently is able to reverse this? So I'm going to the new stuff. I hope you find this interesting. So this is the last part of my presentation. How can we rewire the brain? Is this possible? I have no proof of this, but I'm going to show you some in vitro data that points in this direction. So here what we did was a study in which we extracted stem cells from the brain region associated with memory in mammals. In this case, it was mice. Adult mice. There's a certain rate of birth of new neurons in the adult brain of mammals and of humans and other, of mice in humans, in other mammals. So we induced these neural stem cells to proliferate, and we cultured them in saline and also in the beta-carbolines that are present in the Banisteriopsis capiti. I'm going to show here results for harmine and tetraharmine. And we measured the capacity, if it had any, of these substances to promote the early stages of neurogenesis, of the generation of the birth of new neurons. So this is going to be stained in green, and also the capacity of the substances to promote the maturation of neurons, right? And this is going to be stained in red. So this is the first slide. I used to call this my Picasso slides, but a colleague said, no, it's more Jason Pollock. So these are my Jason Pollock slides for you. Here you have what's called the distal region of the neurosphere. We have a sphere, a floating bunch of neural stem cells, and you have them stained. So in blue you have the cell nuclei. So all cells, all mammal cells have nuclei, so you see all these blue spots, right? In green, you have young neurons. In red, you have mature neurons. So you give them saline, and nothing happens. You do the same experiment with these neurospheres, and you add harmine to the medium, and this is what you get. So you have increased neurogenesis. You are promoting the development of these neural stem cells into neurons young and mature. And the same goes for tetrahydroharmine. So we've already written the paper, we've submitted it to the journal, it's currently under review. There's much more than this, but just as 2 slides as a teaser, I hope you find them interesting. I was really impressed when I saw this. And yeah, just as a final remark, in the psychopharmacological horizon, there has been nothing new for many, many years. So this might have huge potential. Why not look into this? As I say, I think it's a legitimate field of inquiry. We've seen that it already works for depression with rudimentary designs, but at least it shows promise. In addiction, we have the anecdotal reports. Other aspects that my colleagues in the psychiatry department are interested in are psychological trauma. And also, as I said, I collaborate with the neurology department. So could this be of any use for memory loss? We are considering trying these substances in animal models. Neurodegenerative disorders, there are also some animal models that we could use. Alzheimer's disease, Parkinson's disease. And yeah, with this, I finish my presentation. Besides the Beckley, the people in Madrid, there's a bunch of other people who have helped me through the years. I would like to thank them, and also would like to thank you for your attention.
Thank you very much.
You're welcome.
Now, to start off the discussion, the Network for Psychedelic Science have invited Åsa Nilsson, who is a psychiatrist and a senior professor at KI, and also my own mother, incidentally. Please sit down, and I'll turn up the lights again.
Okay.
So let's see, does this work? Yes, it does. So thank you very much. This was extremely interesting. I'm a psychiatrist, which means that not only are we trying to understand what's going on with people, we're actually trying to change the way that they are able to live their lives. Because when they turn up in a psychiatrist's office, there's usually some sort of problem. I've got 3 questions. I'm going to keep them quite short because I'm sure there are lots of incredibly interesting questions in the audience. My first question has to do with addiction and mindfulness. Mindfulness is not actually a treatment as such, but it's a part of a larger philosophical, spiritual endeavor, which has also been used psychotherapeutically, sort of instrumentalizing something which is normally perhaps not primarily but to be used for sort of curing psychiatric diseases or something. Anyhow, it turns out that mindfulness can be very useful when it comes to treating addiction or helping people with addictive problems. And one of the reasons seems to be that there's a narrative in mindfulness, a narrative about what pain is, what suffering is, how to handle the problems that the addicts are trying to handle pharmacologically. Is there a similar narrative in the Ayahuasca movement, or is the treatment purely the ingestion of the substance?
Many of the patients that I mentioned before that as a desperate measure were suggested that they go to Brazil and isolate themselves from their surroundings, be in the jungle for a while, and participate in these ayahuasca sessions, they were very skeptical in the beginning. So they thought, okay, this is going to be a new high, okay, let's go, let's try this. And maybe not after the first session, but maybe after the second, the third, what they report is that ayahuasca gave them the opportunity to reflect on the life path they have been following, and in a very painful and emotional way. So it was quite surprising for them. They didn't expect that. So they were quite stunned. So it was like if someone had slapped them in the head. I call this, I don't know what the exact translation into English would be, but I say that it's like the cosmic slap, right? That you are just on this routine and you just take, and you are with your friends. And the clinicians tell us, we know that they need that something very strong happens in their lives to change. For some people, is perhaps getting a child. The head of the addictions group in my hospital told me that for a person, for him, giving up drugs was associated with 4 years in prison. So you really need a very hard slap to just stop and think and try to follow another course in your life, right? So they said that they had this opportunity, they viewed themselves, the path that they were following, and they could see that it was leading nowhere. And for them it was a very strong motivation, and they were able to do this. So then when they came back to the usual environment in Barcelona, temptations, friends, the usual clique. Maybe some of them got offered again, snort, heroin, whatever. They say, okay, and I ask them, how do you manage to do this, to deal with this? And I say, okay, every once in a while I go to an ayahuasca session, and that reminds me of the path I've selected, and I try to stay away from the substances. So that was quite impressive. I don't know of any other psychopharmacological agent that's able to do this.
I think it's been described also for mescaline.
For what? Sorry.
For mescaline.
Oh, mescaline.
That people who've been addicted to other kinds of drugs have exactly what you're describing, that you have a sort of flash of insight as to where your life is going, and that you really should change path.
I think it's the key. There's a cognitive effect. There's the insight that these substances provide. And in the US, there's been a couple of studies done with psilocybin and tobacco addiction, for instance, and the results were impressive.
So basically, it comes from inside. So that was my first question. And the second question was, when it comes to mindfulness, for instance, there's been a lot of interest in trying to understand what goes on in the brain when people train mindfulness. And one of the interesting things to come out of that is that it seems that there are differences between men and women. For instance, in the way that the hippocampus is activated during different kinds of mindfulness exercises. And I was wondering about your data. You didn't have information about men and women. And do you think it would be interesting to separate them, as perhaps these mechanisms would be different in men and women?
Thanks for the suggestion. The studies we've done so far have been with small samples.
Yeah.
So maybe we wouldn't have the power yet to separate or to look for gender differences. But we are getting more and more data coming in. So we should definitely look into this.
So my next question is a very broad one. You were talking about the intimate aspects of the human psyche. And if you're describing the effects of these kinds of drugs as being profoundly meaningful for the people who are experiencing them, how do people react when you tell them that this profoundly meaningful experience comes down to an activation of the anterior cingulum or top-down disturbance, the frontal lobes being sort of shut down? Are you accused of being reductionist sometimes? And how do religious people respond to this? Because there's a spiritual dimension to this also.
Yeah, I'm not being accused of being reductionist sometimes. I'm always being accused of being reductionist. And I don't try to, feed my jargon into other people's throats. It's just my approach, and sometimes in my presentations I use a slide in which you have this elephant and blindfolded individuals with wearing white coats, and each has a different view of what an elephant is. If you're touching the tusk, or you're touching the trunk, or you're touching the leg, or you're touching the tail, right? So it's just an approach. It's our approach. We have these tools. We can measure what we can measure. Of course, it's just one aspect of it. Some people view it as a religious experience, as a mystical experience. And from what we see is that depending on the assessment tool you use, if you use EEG, you can see some aspects of changes in the brain. If you use SPECT or fMRI, you see something different. So nobody has the whole truth. And I really respect their views, so I'm not trying to convince anyone. It's just that I try to see if there's something going on in the brain, because this is my field of interest. I try to understand this in biological terms, but by no means I think this is all that there's to it.
In any case, it's very, very encouraging for psychiatrists to get a broader view. The man with the telescope was sort of, I think he's going to see something very soon. And I'm sure that we really need more information, and as clinicians, about the processes that you're studying. So thank you very much. And so we'll just leave the questions to sort of pop up by themselves.
Yeah, just as a comment, there's also the language in which you have to transmit the information that you obtain to your colleagues, and this is the language they speak. They speak the language of brain maps, with these fancy colors and these unpronounceable names, right? So if you only get the subjective reports and you say, okay, my subjects experience a mystical union with the universe, it will be difficult to convince them that maybe this should be studied in pathological conditions. You say, you know what, I saw these changes in emotional processing areas, then they start paying attention. So it also has this advantage.
I actually can't stop. I just have to give us this last comment. Maybe these images are going to be sort of like the Rosetta Stone, because I think there's a difficulty, because we have one terminology in psychiatry, we have one terminology in the religious groups, we have one terminology in the people who use drugs, and maybe the image of what's going on will make it possible for us to communicate much better and to sort of have a dictionary so we can communicate more freely.
Yeah, we're working on a puzzle and each of us tries to put some piece in it.
Yes. So thank you.
You're welcome.
Thank you so much. Now we have time for 1 or 2 questions from the audience before we end. Our time is up. It's been a great pleasure to hear your talk and the discussion.
My pleasure to be here. Thanks for your patience.
Let's thank Jordi Riba and Åsa Nilssonne, and thank you all for coming tonight.